Okay, so good afternoon, everybody. I'm Lucy Fairclough, Professor of Immunology and Chair of the Midlands Innovation Extracellular Vesicles Network. And so I just thought before we start the afternoon, I'll say a few words on behalf of the Midlands Innovation EV Network. So, as I'm sure you all know, extracellular vesicles have fundamentally changed how we think about how cells communicate. So, we used to think about them as byproducts of cellular activity, but we now know that EVs are recognised as important carriers of biological information. And amongst their most interesting cargo is RNA, which is diverse, dynamic, and really highly informative. So studying the transcriptomes of extracellular vesicles offers us a unique perspective on cellular state, communication between cells, and potentially disease processes that can't be captured by normal traditional cell-bound transcriptomic analysis. But again, as we all know, EV transcriptomics gives us really distinct challenges. I think the first one, which the first session is going to address, is low input material. Another challenge is the fact that cells produce lots of extracellular vesicles, but we know there's a lot of heterogeneity between the populations of vesicles within any preparations that we might prepare. We've also got the challenges of isolation strategies, from using size exclusion chromatography through to using ultracentrifugation. There might also be things like sequencing biases that we need to think about. And then the biggest challenge at the end is probably data interpretation. But all of these need us to ensure that we think really carefully about the experimental design and analytical choices that we make when we're looking at EV transcriptomic analysis. And basically what I've just introduced are all the things that we're going to be talking about in this afternoon's networking session. So we've designed the workshop to hopefully include a practical perspective, so thinking about what sorts of things you need to think about if you want to do EV transcriptomic analysis. We're hoping it's going to be interactive, so we really want to hear what your experiences are, what your views are. It's not just us talking to you, it's also you talking to us and telling us about your experiences. But over the course of the afternoon, we're going to think about different methodological advances that have occurred in the field, different approaches to analysing EV data, and also thinking about biological insights that we can draw from any EV RNA data that we can generate. But like I say, although it's us talking, really just as important is for you to talk to us. So we're going to have opportunity about discussing what does work, but also probably even more importantly, what doesn't work or what challenges you found working in this field. So please do ask questions and please do share your experiences, whether they're good experiences or they're challenges that you faced. Progress in this field is really important, and it really depends on us working together and using collective learning and being transparent with any of the challenges that we faced. Again, I'm hoping we've got some early career researchers and students on this call. So this is really an important evolving area. And again, we'll be really welcome any questions that you might have, perhaps new coming into the field, because your insights will be really, really helpful to us think about the things that we need to address going forward. And also potentially any other future training opportunities that we might want to put on as part of the Midlands Innovation EV Network. So before I hand over to our two speakers, I'm going to thank all of the speakers today for contributing both their time and expertise. We hope that this workshop does spark new ideas. We hope it also strengthens collaborations, both within the Midlands, but also externally, because I know there's quite a few external people on the call today. And most importantly, I hope it moves this field forward. So with that, I'm going to hand over to our first speakers for this afternoon session, and that's Dr. Josh Price and Dr. Georgia Hopkins. And they are going to talk to us about how we prepare samples for doing EV transcriptomic analysis. So thanks very much, Georgia and Josh. I'll hand over to you now.
Hello, can you hear me and can you see my screen, first of all?
Yes, and yes. Thanks,
Yeah.
Josh.
Perfect. Thanks for the introduction, Lucy. Georgie and I have made this presentation together, and I'm going to go over some tips, tricks, and things to think about before you even start your experiments with extracellular vesicles, and Georgie's going to take over getting into the RNA part of it as well. So, we've got two major aims, so we've split the presentation in half. We want to understand when to perform EV RNA analysis, the challenges associated with EVs, with getting hold of EVs and then getting hold of their RNA as well, which is aim 2. So we'll cover an introduction to extracellular vesicles, typical EV preparation strategies, and maybe a little bit on RNA quality control as well. So the first question really is, what are EVs and the associated challenges with RNA analysis? So the MySev answer to that, our guidance from the minimal information for standards of extracellular vesicles, is membrane-bound nanoparticles that cannot replicate. They're released by cells, they contain proteins, lipids, and very importantly, RNA. There's a little bit of history. They were described as early as 1946, and ultracentrifugation was found to isolate them. So you can see that in platelet-free plasma, clotting occurred, but when we pelleted the particles that were responsible for that clotting, at the time called platelet dust, we realized these now to be extracellular vesicles, these procoagulant particles. We now recognize them to be various shapes, various sizes, going all the way up to potentially 500 nanometers with these apoptotic bodies, but as small as 40 nanometers as well. They're found in all biological fluids. They're found in plasma, in urine, in BALF. They're released by all cells, and importantly, they contain microRNA. We split them broadly into those of endosomal origin and those of plasma membrane origin. And so when we say endosomal origin, the other thing you might see in the literature is exosome. And for plasma membrane origin, we're thinking microvesicle and apoptotic body. Despite this versus of endosomal versus plasma, it's actually very difficult to tell them apart because markers of their origin overlap. So we tend to look for things like CD81, CD63 and CD9, and these indicate to us that we're likely dealing with an extracellular vesicle sample. So why study RNA as a specific cargo of EV? And one of the major reasons is they're important for intercellular communication. MicroRNAs can regulate multiple genes, multiple pathways in recipient cells, which can lead to changes in metabolism, in inflammation, and can change the function of cells. And failing this intercellular communication and gene regulation role, they can also act as disease biomarkers, where they change with obesity, they change with cancer, and they change with other disorders as well. So how do we determine whether RNA-seq is appropriate and whether it's EV RNA-seq that we need? We have to start with asking a couple of questions. The first one is, does the biological question require EV-specific cargo? Potentially, we're interested in what EV do to cells, in which case we could do an experiment where we treat our cells with EVs and instead sequence those cells, and that might be an easier approach that answers your question more directly. We can also ask the question, are EVs the thing that is causing the phenotype that we observe? If we're treating cells with a secretome, maybe we could look at extracellular RNA instead of extracellular vesicle RNA. And the other important factor that we'll touch upon in more detail later is whether we can isolate sufficient EV material to justify collecting RNA and doing RNA sequencing analysis. So it's very important that we answer these questions and justify early in our experimental design whether it is EV and EV RNA that we are interested in. So in a bit more detail, the differences between the two, when we look at EV cargo, we tend to be looking at RNA in EVs, and that's typically microRNAs, and that's because most of the EVs we look at are between 40 to 200 nanometers in size. They contain mostly small RNA fragments and microRNAs, and microRNAs are only 22 to 24 nucleotides long. We tend to get very small amount of RNA from EV RNA isolations, and some of you may be familiar with this term RIN, which means RNA integrity number. This is often not meaningful when we're looking at small RNA in vesicles because it relates to ribosomal RNA length, and that's not present in a vesicle sample. So we use EV cargo sequencing when we're interested in typically the microRNAs that are in the vesicle. And this is really useful. It gives us a view of what's inside the vesicle, and it can be useful for biomarker discovery. Often cancer cells will be releasing microRNAs that tell us something about what the cancer is doing. But we are limited with material, and we don't always have a guaranteed function in recipient cells just because our EV contain this cargo. On the other side, we have our EV treated cell sequencing. So this is more typical RNA sequencing, where we're looking for full length mRNAs typically. We can get good amounts of RNA from basic methods. RNA integrity number, or the RIN, really does matter here. We want to make sure we're dealing with RNA that's of high quality. And it's really, really useful when we want to see what EVs are doing to our recipient cells. So we've covered this a little bit. So our EV RNA characteristics, they are low yield, highly fragmented if they are mRNA, but our microRNAs would be in this region too of 22 to 24 nucleotides. The RIN value that we'd be looking for is sometimes 0, but often between 2 and 3, and it's all microRNAs and tRNA fragments. What this means is we have to use small RNA sequencing protocols. Specialized alignment is needed. We can't use HiSat2, for example. We're better off using things like Bowtie or MerDeep, and we have to trim our adapters quite sensibly because they're going to be in a lot of our reads. Once we get to the stage we're looking at microRNAs that we're interested in, we can start looking at target databases, and the best information I can give you there is to look as many different databases as you can and look at the overlap. And when we're doing our isolations, include C. elegans microRNA typically, because we can use this as a quality control across our different samples. The final thing to think about from my point of view is where we're getting our EVs from. Are we getting them from cell culture? If we are, we have to be careful and not use fetal bovine serum. Fetal bovine serum is typically used in your cell culture medias, and that will contain vesicles, and it will also contain vesicles that contain RNA. So this will be a problem for our downstream analysis. So we have to use serum-free media or FBS-depleted media, which means sometimes the vesicles we'll be looking at, we have to bear in mind, might be because of stress. When we think of plasma and serum, we have to think about our lipoprotein contamination. We get both HDL and LDL in there, and they can look very similar to vesicles, and a lot of our isolation methods will co-isolate them. Platelet-derived EVs can be a real challenge here as well. If we don't fully remove our platelets, normally through double centrifugation, we end up with an artifactual increase in the number of platelet-derived extracellular vesicles. And it can be quite a complex RNA background in plasma and serum. When we get to bronchoalveolar lavage fluid, typically we get low EV, can be quite difficult to collect, and we get mucus and protein contamination. All of this changes the protocols that we're going to use to isolate them, and there's lots of good reviews out there, so I won't go into detail on how to do that, but you really need to consider what isolation protocols you're going to use ahead of time. And so with that, I think I'm going to hand over to Georgie to take over the rest of the slides.
Perfect, thank you, Josh. Okay, can you see that?
Yes.
Okay, so now Josh has gone through with you about whether to go ahead with RNA analysis of your EV samples, and you've thought about some of the associated challenges, then you can actually begin to start preparing your samples for transcriptomics analysis. So before we even talk about EV isolation or RNA isolation, we need to talk about the initial sample collection, because pre-analytical variables are one of the biggest sources of inconsistency in EV studies. So after you've collected your sample, whether it was plasma or your cell culture supernatant or your BAL sample or urine, you need to remove your contaminants as early as possible. These can actually impact your downstream data by releasing RNA and forming EV-sized fragments after processing. And actually, the longer they stay in your sample, the harder they are to separate. So as Josh just mentioned, you have contaminants in your plasma or blood, like including things like platelets, or in your cell culture supernatants, you might have some contaminating cells or cell debris, or protein aggregates, and in BALF you might have mucus and protein complexes. But you can remove a lot of these by centrifugation. So you can do some low spins to get rid of cells or mucus. You can do 2000 G to get rid of cell debris, or you could do a 5000 G for platelets, or some people do a double 2500 G spin. And then some labs actually use micron filters to remove cells or platelets, but these can actually cause issues because they aggressive filtration can actually reduce your yield if you're not careful with them. And if you're using cell culture supernatant, like Josh just said, remember that serum can cause contaminants, and so you need to use serum-free media or EV-depleted FBS if possible. And you should also perform a live cell count, especially in cell culture supernatants. So especially if you're comparing conditions, make sure your you need to make sure your EV output differences aren't simply due to differences in your cell number. So always normalize to cell count or protein content where appropriate. And another key point is the starting volume. So higher starting volume, as you would expect, usually improves EV yield because you'll have more EVs. And the storage really matters as well. So repeated freeze-thaw cycles will damage vesicles and degrade RNA. And so the idea is to process your samples really quickly, keep them on ice, minimize the time between collection and isolation. Because ultimately at this early stage, consistency is really important, and you need to do all the same steps for all your samples, because if you start to get variability at this early point, no amount of downstream quality control can actually fix it. And so once you've collected your sample and done some pre-clearing, you can then move on to isolating your EVs. So there's no universally best method for this. It's only what is best for your biological question. But a common method is ultracentrifugation. And so this involves sequential spins at increasing speeds, often finishing around 100,000 G or higher. But it can co-isolate protein aggregates and lipoproteins, especially if you're using plasma samples. So that's something to bear in mind. But you can also do size exclusion chromatography, or SEC, and this is becoming increasingly more common. It separates particles by size, it's much gentler, so it means that you maintain vesicle integrity and you reduce the soluble protein contamination. But it, like the ultracentrifugation, it also doesn't fully remove lipoproteins in your plasma, and yield can be slightly lower depending on how you pull the fractions after size exclusion chromatography. So they're the two main techniques that people use, but there are a few others that you can bear in mind. So you can do density gradients, and these improve purity further by separating particles based on density. And they're really powerful, but they are time consuming and technically demanding. And then there's polymer-based precipitation. So it's simple and high yielding, but it co-precipitates contaminants. So for RNA studies, especially biomarker work, this can really confound your results. So that's something to bear in mind. You can also do immunoaffinity capture, which allows you to isolate specific EV populations using antibodies against markers like CD63 or CD81. So this is great if you want to do a targeted mechanistic study, but obviously it is biased because you're selecting a subset of EVs, not the total EV population. And then finally, you've got microfluidics-based systems, which are emerging technologies, and they can integrate isolation and analysis, but they're often still optimizing for reproducibility and scalability. And so with all of these methods, there's trade-offs between yield and purity, and isolation methods, they directly shape your RNA profile depending on which one you use. So if you change isolation strategy mid-project, your RNA data may change even if the biology hasn't. So my tip there is to choose your isolation strategy early and stay consistent. So then once you've isolated your EVs, you can move on to concentrating them and some of the quality control steps. So after methods like size exclusion chromatography, you can then concentrate the EVs afterwards, because you end up with like 5 ml of volume. And for transcriptomics, you want quite a small volume of really concentrated EVs. So we use Amicon centrifugal filters at 10 kDa, which then leaves you with about 250 microliters afterwards. And then once you've concentrated them, or you're happy with the volume you've got, you need to characterize them, particularly for particle count. So typically people use nanoparticle tracking analysis or tunable resistive pulse sensing, and that gives you an estimate of how many particles you have and their size distribution. And I might be biased here, but I love flow cytometry, and it's also a great way to quantify your EVs in your sample. So you can actually stain for dyes such as calcium AM, which only fluoresce when inside an intact EV. And so then you only get an EV count for intact EVs. Particle count is essential to make sure that they're concentrated enough for transcriptomics. And then some people do further characterizing of the EV sample. So you could look at protein markers on the surface like the CD63, CD81 and CD9. But equally, it's important to look for the negative markers like calnexin, which should not be present in a clean EV prep. And this is where I strongly recommend following the MyCEF guidelines by the International Society of Extracellular Vesicles, which Josh mentioned earlier. And they don't tell you exactly how to isolate EVs, but they provide a framework for reporting and characterizing your EVs, which is really important. So this is just an example of a workflow that we've done in our lab, and it's a really simple workflow from sample collection all the way through to transcriptomics analysis. We used human bronchial epithelial cell supernatant. So we would take out the basal supernatant from the cell culture well, and then we'd do our pre-clearing first. So we would spin at 300 G, followed by 2000 G spin to remove the debris. And then we apply the supernatant to a QEV original 35 column from ISOM. And then we use automated size exclusion chromatography with this machine from ISOM. And so the EVs then are isolated into 7 different fractions, depending on their size. And then we ball all of those 7 fractions together to then concentrate them using the 10 kDa Amicon columns, which results in about 250 microliters of EVs in solution, ranging in size from 35 nanometers to 350 nanometers. But then we check the particle size and their number using nanoparticle tracking analysis. So then we can freeze our samples at -80 at around 10 to the 8 EVs in 200 microliters of volume. And then we would simply just ship the EVs off to our sequence provider for microRNA cargo analysis. So this is just an example of a really simple workflow, but importantly, this is to result in just EV samples that you're shipping off. So this hasn't included extracting RNA from your EV samples. These samples will be sent to the company, who then perform their own standardized RNA extraction techniques, which can sometimes reduce variability between your samples. But if you don't want to do that and you want to extract your RNA yourself, there's a few tips here. So just remember that you're working with extremely low input, so the recovery efficiency matters enormously. And we strongly recommend column-based kits. So they're designed specifically for low input or microRNA samples. And avoid phenol or chloroform to purify RNA unless you're very confident, because at sub-nanogram levels, recovery becomes inconsistent and phase separation losses are magnified. Always include on-column DNA's digestion. So even in tiny amounts of contaminating DNA, these can distort downstream quantification. And this might sound obvious, but also use RNA's free buffers throughout. But if you might think in shared lab spaces, sometimes contamination can happen. So just bear that in mind. And as Josh said earlier, it's super duper important to include your spike-ins as a control. So exogenous spike-in RNA, like C. elegans microRNA, at a fixed amount during lysis needs to be used as a control. And that allows you to monitor extraction efficiency and technical variability independently of biological EV RNA content. And then in terms of expected RNA yields, so EV pellets typically contain between 0.5 and 5 nanograms of total RNA, which is not much. So you will not see ribosomal RNA peaks, but you will see enrichment of short non-coding RNAs such as microRNAs, yRNAs, transfer RNA fragments, and yields vary hugely depending on cell type and isolation method. So always pilot before committing pressure samples. And it's best to design your experiment assuming yield will be at the lower end of what you hope. So in terms of the minimum input requirements, because of these low yields, you must use small RNA-specific library prep kits. And inputs as low as 1 nanogram can work with specialist kits, but you need to plan carefully. And size selection for inserts between 80 and 40 nucleotides is critical. Otherwise, you'll sequence adapter dimers. Also, unique molecular identifiers are very helpful at low input, and they can reduce duplication bias. And then finally, don't use messenger RNA sequencing kits because they simply won't capture the small RNAs enriched in EVs. And at this stage, if you use the wrong kit, you'll almost get no meaningful data. So it's really important. So once you've isolated your RNA, you need to quantify it. We don't recommend nanodrop because at these concentrations, you're essentially going to be guessing the concentration. But you can use qubit fluorometric quantification, and it's sensitive down to low picogram to nanogram levels and uses RNA-specific dyes. There is minimal interference from protein or phenol, and it's much more reproducible. But you can also use RNA nanochips for long RNAs, which might not really be relevant. So most people actually use the small RNA chips for shorter RNA, which also measures the RNA size. So essentially, you match the quality control tool to the molecule type that you're expecting. And finally, what actually matters in RNA quality control? So first, the qubit concentration definitely matters. That's your reliable quantitative measure. And secondly, the small RNA profile. Do you see enrichment in the 18 to 30 nucleotide range? That's really important. The spike in recovery that we've emphasized a few times. If your spike-in isn't detected at expected levels, something went wrong in the extraction or your library prep. And then fourth, the library fragment distribution after prep. So you should see a clear peak corresponding to a small RNA insert plus the adapters. And finally, the sequencing read length distribution. So if most reads are adapter only, your size selection probably failed. But you need to interpret QC as part of a story, not as a binary pass-fail situation. And then in terms of what doesn't really matter, like Josh said earlier, the RIN scores are meaningless really for EV RNA. There's no intact ribosomal RNA, so the algorithm assumes degradation. And absence of RNA peaks, the ribosomal RNA peaks is completely expected. The bioanalyzer total RNA quantity below 5 nanograms is also unreliable. So if you evaluate EV RNA with tissue RNA criteria, you'll think every sample is terrible, but it's not. It's different biology, different rules, and you just have to bear this in mind when you're going from cell RNA analysis to the EV RNA analysis in QC. So key takeaways. First, you need to confirm that RNA analysis is actually appropriate for your biological question. And then you need to understand the biological source and its contaminants and the challenges associated with this. And then you can move on to your preparation of your sample and controls to determine success more than sequencing depth. You always need to assume low input and bias. Recognise that your isolation method shapes your RNA profile. So remember that SEC or ultracentrifugation or precipitation, they're all different methods, they're not interchangeable. And quality control is about informing interpretation, not necessarily rejecting samples unnecessarily. So that's the end of the first session. We're expecting a lot of EV transcriptomics newbies on this call. So feel free to put any questions you might have in the Q&A, or you can turn your microphone on or your camera and ask us anything you like. No question is a silly question. So thank you.
Thanks, Georgie and Josh. That was great. Really great first session. So yes, has anybody got any questions that they want to ask George or Josh about preparing samples for sending off for transcriptomic analysis? Can't see anything in the Q&A at this stage.
We can always put them in the chat and answer them later as well, you know, give people time to think.
Yeah.
Well, there's a question here from somebody. So with sequencing EV treated cells, is microRNA still detected if is messenger RNA, should read this carefully, Lucy, is messenger RNA still detected if EVs derived microRNAs are transcriptionally repressing the the messenger RNA rather than directing the messenger RNA towards degradation? I'm not sure if I've read that question quite properly.
Let me have a look as well. I think you're getting at whether the mRNA will still be there if our microRNA are acting upon them. For the most part, it will depend on timing. So I would say yes, but it will be a matter of getting your timings right as well. So if you do a couple of different time points, hopefully you'll see that. But it wouldn't make, at that point, wouldn't put me off doing it. I'd still trust the RNAs to be there, but yeah, I think it would be fine to do that.
Okay, thanks, Josh. Anybody got any more questions before we move on? No? Well, thank you very much again, Josh and Georgie. Really great first session. So we will now move on to the next session, which is from our commercial companies who are going to talk to us about doing RNA analysis. So, Matthias, are you on the call?
Yes, I'm here. Do you hear me?
I can hear you. Thank you. Are you okay
Perfect.
to share your screen for us?
Sure. Yeah, let me try.
We're 5 minutes ahead of time, but we may as well. That's completely fine.
Okay. Good. Do you see my screen?
I do see your screen.
Yeah.
Lovely.
Okay.
Thank
Excellent.
you. So about sort of 10 or 12 minutes, as we're slightly ahead, you have actually got an extra couple of minutes if you need it.
That's excellent. Yeah, I may need it. Yeah, so we'll
Okay.
see how it goes. But yeah, thanks, Lucy and Georgie and everyone, actually for giving us also the opportunity to present here. So I'm Matthias. I'm a co-founder and CSO of a company called Tamirna. Okay, yeah, here we go. So Tamirna, for those of you who don't know us, we are based in Vienna, in Austria, and actually originally we and this is still one of our main objectives, but the idea for Tamirna is to become a diagnostic company and a developer of in vitro diagnostic solutions, and doing this based on detection of microRNAs as biomarkers. And we've been doing this now for more than 15 years. With a few successes, we got our quality management system certified. We got our regulatory in place. We also got our science in place. So we found a good biomarker for liver function that we ultimately took through regulatory approval. And I think we're now one of the first manufacturers of an IVD test that is based on detection of microRNAs in a liquid biopsy. All these activities and all that R&D also led to another sort of exploitation of our science, which is services. So a while ago, I think six or 7 years ago, We actually been asked if we could perform RNA biomarker analysis for different customers, focusing on microRNAs, but also long RNAs, but always focusing on liquid biopsies as they have an advantage in diagnostics. And that at some point about 5 or 6 years ago, then also started to include extracellular vesicles as one of the potential carriers of cell-free RNA. And that's where our expertise in that area also grew a lot. So what fascinates me, and also as a diagnostic company, is the visualizing or envisioning vesicles as carriers of messages, like a message in a bottle. And that cargo or that message can inform us about the physiological state of a cell and ultimately give us information about biological potency and efficacy of EVs. Or that cargo can also inform about the pathological state of the donor cell, and ultimately that can be really useful to develop new biomarkers. The question is only how can we access that message? And Georgie gave already a really nice introduction about RNA and some of the constraints, and I'm going to follow up on this from the RNA perspective. And I'm kind of repeating what has been said before, but we cannot apply quality control metrics of tissue RNA to extracellular RNA. That's one of the first learnings that we also had to do, right? So all of these things like RNA integrity value or photometric scores on purity cannot be applied because the reality is, as has been said before, extracellular RNA is short, longer RNAs are fragmented, you don't see ribosomal RNA, and so we're a little bit tapping in the dark when we are looking at these electropherograms, whether the sample is of high quality or not. But when we spend a lot of money on sequencing, we want to make sure that actually we're knowing what we're doing and that we really understand the quality of the data that we are producing from these kind of samples. Spike-ins have been mentioned as one of the options, and I'm also fully convinced that adding exogenous RNA to your sample at different stages in the process can be really useful. We took this a little bit further and developed our own spike-in cocktail. We call them the MIND spike-ins. And that's actually a mixture of 7 different oligonucleotides, and they span a concentration range that overlaps with endogenous RNAs. We add them to the sample prior sequencing and then construct libraries and analyze the data, and that workflow is actually fully published also. It's been developed as part of an EU collaborative project that we participated in. These spike-ins, beyond being 7 different spike-ins, they also have been optimized to a very low sequencing or ligation bias. So we really, whatever we add is ending up truly represented in the sequencing library. So long story short, what we actually get with every sample that we sequence from our own projects and our customers, we get this type of QC where we can first of all confirm the dynamic range of the sequencing assay. We plot the lowest to the highest abundance spike in the read count against the copy number that we put in, and then we can overlay the dynamic range of spike-ins with the dynamic range of microRNAs that we have detected. These are the dots you see here on the right, and this is for us really the best way of actually interpreting RNA yield and RNA quality, but also sequencing quality. So that is really peace of mind for us. It also can help to evaluate batch-to-batch variation using these spike-ins. You can actually also derive absolute quantities of microRNAs, relate this back to your EVs you put in, and sort of have a more absolute analysis of microRNA copies. And you can use these spike-ins also to benchmark your small RNA sequencing workflow. To English, we actually done this. So we just recently benchmarked, I think, 6 of the most widely used small RNA sequencing library prep techniques for low RNA input, from single adapter ligation protocols to dual adapter ligation protocols using UMIS. And we see that these spike-ins are really compatible with any of these workflows. Once you have generated your data, one other key or the next key step is actually interpreting that data. And that can be quite tricky. So what we've been doing in Tamirna for the last years is also spend a lot of time, and you can see a little movie showing our reports, to turn raw NGS data into a report that even non-bioinformaticians like myself, we can work with, we can interpret, and we can get a deep dive into our data. We call this the MIND report. That is usually also included with the analysis that we do. Also, how we generate this is fully published. You can download the paper. You can also download a demo report from our website, where it is explained, all of the, you can see all of the analysis that we do from QC to actually quantitative information, such as heat maps, principal component analysis, differential expression analysis. And what we also added additional tools, for instance, to include target prediction of microRNAs of interest, impact on biological pathways. We have tools to look at microRNA isoforms, which can also include information, biological or pathological information on your system, or even look at other types of small RNAs using a third sort of add-on pipeline that we developed. So maybe to put this in a bit more biological context, I've prepared 2 case studies that I would like to briefly go through. So the first one is really how reproducible is small RNA sequencing from EVs. And to address this, we've worked with a company called Rooster Bio. And we published this white paper that you can also download from our website. So essentially here, the idea was Rooster has a biobank of extracellular vesicles that they have produced from mesenchymal stromal cells or stem cells. And these stem cells have been isolated from different original tissues, umbilical cord, adipose tissue, and bone marrow. And we wanted to understand how reproducible are different lots of their sort of off-the-shelf EVs, but how reproducible is also our analysis. So we teamed up and essentially analyzed those in technical replicates. And what we see is that the analysis itself, when you correlate technical replicates, is highly reproducible. And we can show this here based on EVs from bone marrow, umbilical cord, and adipose tissue. And then when we look into these profiles of these, EVs from different tissues of origin, we can also see that origin highly impacts the microRNA profile. So we can see clusters only resembling umbilical cord-derived mesenchymal stem cell EVs, adipose tissue, and bone marrow. And you can also see that this is actually also highly reproducible. So we have very stable RNA expression patterns. And we also saw that some microRNAs that are known to mediate inflammatory responses, such as microRNA 121.5p, 146A5p, that control NF-kappa B signaling, for instance, are actually also differentially present in these different EV fractions, which may have an implication on their biological potency. I think I have a bit of time left, so quickly another case study that we recently contributed to. This is from a company called Extracellular, and this has just been accepted in Stem Cells Translational Medicine. So what they are doing, they're interested in using their EVs to treat neurodegenerative diseases. And they had a certain manufacturing process, starting with a 2D culture to ultracentrifugation-based isolation. But they realized at some point that's not scalable, so they switched to 3D cultivation, actually TFF, tangential flow filtration, and SEC to purify. And really the idea was to use multi-omics analysis to understand what is the impact of switching your process of manufacturing from and purification, especially from ultracentrifugation to TFF-SEC. And so we obtained their EVs and we did small RNA sequencing for microRNAs and whole transcriptome sequencing for mRNA. In a nutshell, what we observe is that the microRNA signal is more dependent on purification than the mRNA signal. We've seen that going from ultracentrifugation to TFF-SEC, we have a drop in the number of microRNAs we can detect. And also we see less correlation between those 2 types of purification methods in the microRNA profiles, while the mRNA profile was actually less impacted by this scale up. And so what we hypothesize is that certain non-EV impurities that may be present with ultracentrifugation that you may be able to better remove with SEC are actually also loaded with microRNA, and that give this additional background in an ultracentrifugation-based fraction. What we also did in this project, I'll be really short here, we also analyzed how the microRNA EV the EV microRNA cargo could actually interact with the target tissue in this neurodegenerative disease model. And for this, we used actually RNA-seq to look at mRNA expression after EV treatment. And we found that there were some really nice interaction between some of the highly loaded or highly enriched microRNAs in these EVs and some of the pathways such as cognition that were impacted in treated animals. So to summarize this, what we can do, as a service provider, we can in extracellular vesicles, but also cells, tissues, and liquid biopsies, we can analyze microRNAs and mRNAs using different RNA sequencing methods. I only focused on the MIND, but there's another one that we have for long RNAs. What's important to us is that we actually always start the project with a scientific consultation, because every project is different. There are different prerequisites, and we really want to understand the aims and goals of our partners before we go into a project. And just some logistics. Yeah, we can work with total RNA or EVs. You know, pros and cons to both, as Georgie also pointed out. We like to be challenged. So if specs fall below our standard requirements, we can always run a pilot to determine feasibility. We can organize sample pickup, race and it usually take about 4 to 6 weeks to complete this analysis for our customers. So yeah, I hope I could tell you a little bit about Tamirna, what we're doing. We are a small team here in Vienna, all scientists, and we'd like to approach projects really on a scientific level. So if you have any questions or needs, just feel free to reach out to us anytime. Yeah, back to you, Lucy.
Lovely. Thank you very much. And you kept perfectly to time. So thank you very much, Matthias. I think what we're going to do is move on to the next commercial talk, and then we'll take questions for both of you at the end, if that's okay. Thank you. Okay,
Absolutely.
so next I'd like to invite Quinn Zhang, who is the project manager at BGI Genomics. If you're on the call, if you could share your screen, please, that would be great.
A minute. Do you see my screen?
Yes, if you could just put it on presenter view so we can see just the perfect. Yep, that's great. Thank you very much. Okay, so just over 10 minutes, if that's okay.
Okay, thanks, Lucy.
Thank you.
Today, I'm excited to share our high sensitivity small RNA sequencing solution for EVs. Exomes are special subtype of EVs. Exomes are normal-sized membrane particles secreted by cells to transmit information from cell to cell, and can be found in almost all body fluids, including blood, saliva, or urine. And the exome research now has a wide range of applications. That leads to the question: what do we study in exomes? It really depends on where the exomes come from. Different origins mean different cargo, lipids, protein, RNA, and DNA, each with distinct functions. Among these, long-coding RNA have rarely taken center stage as one of the most active research areas. And non-coding RNAs are where the real action is. In eukaryotic genomes, protein coding genes account for only about 1 to 2%. Long-coding RNAs regulate gene expression at multiple levels: transcriptional, post-transcriptional, and epigenetic. Now I'd like to introduce our small RNA sequencing solution. Small RNAs are important functional non-coding RNAs molecules that are less than 200 nucleotides in length, include microRNA, cRNA, and piRNA. They act as sequence-specific sensors to govern development, stress adaptation, and genomic stability. DB6 small RNA sequencing mainly focus on microRNA with part of cRNA and piRNA. The technology enables the profiling of 18 to 30 nucleotides and 15 to 30 nucleotides, small RNAs in specific tissues and cytological states across species. The DNB6 small RNA sequencing consists of 4 main steps. Suitable samples can include total RNA, tissues, serum and plasma, FIP, exomes, and RNA. Then we use the UMI technology directly into library preparation, and that's what makes ultra-low input possible. Since mRNAs are short by nature, we recommend single-end 40 base pair sequencing with 20 million reads, clean reads, or more per sample. We also provide professional bioinformatic analysis. And for spatial samples, we recommend at least 5 nanograms of total RNA as input. Okay, let's go deeper into our library preparation solutions. At BGI, we offer UMI-based mRNA libraries, and we also have a dedicated low-input workflow optimized for challenging samples. And for spatial samples like exosomes, we extend the insert size to 15 to 30 nucleotides. That gives you broader coverage of the small RNA landscape. So what exactly is UMI? The unique molecular identifier is a short sequence of 8 to 10 nucleotides, which can be regarded as a barcode. During library conjugation, is ligated to cDNA molecules to label each molecule in the sample, ensuring that each small RNA is labeled with one type of UMI. Why does that matter? Because it effectively frees the pre-amplification state. We can trace each read back to an original molecule, recover true quantitative information, and strip away the base introduced by PCR amplification. That's a game changer, especially when you are working with low input samples where every molecule counts. So what can we get with this technology? More accurate, low input, high reproducibility, and repeatability. UMI technology boosts biological reproducibility increased by 15%. The starting amount of the sample can be as low as 1 nanogram. We also offer comprehensive bioinformatic analysis for small RNA sequencing. The standard analysis pipeline includes microRNA expression, microRNA differential expression, microRNA target genes, KGG enrichment, and the analysis results are packaged and uploaded to the Dr. Tom system. This system is our proprietary interactive data mini platform. Dr. Tom pulls in multiple public databases and some analysis tools. We can identify long mRNAs, microRNAs directly through the system and run flexible interactive analysis without writing a single line of code. We can also offer BGI report system, which has low spatial limitation. As long as you have a reference genome or a gene set, we can predict a low-vol microRNA, cRNA, piRNA. And if your project has specific leads, we can also support customized bioinformatic analysis. Okay, let's move on to the advantages of DNB6 small RNA sequencing. We have UMI small RNA library technology, which enables high accuracy for quantification. DNB6 sequencing platform, which enables lower duplication rates. Dr. Tom system, which enables data analysis results, visualization, and interpretation feasibly. With extensive experience, we process around 8,000 to 10,000 samples every year. So what you get is a solution that is cost-effective, highly accurate, high reproducibility, and high compatibility. As of December 2025, we have supported 500 publications with a combined impact factor of over 3,000. Finally, let me briefly introduce BGI's one-stop multi-omics solutions. We offer a broad and integrated services portfolio, including gene synthesis, and DNA, RNA, epigenetic, single-cell, and spatial transcriptomics, microbial research, and proteomics, metabolomics. With decades of experience in research services, BGI is fully committed to empowering life science discovery. We also have the Dr. Tom multi-omics data mining system, our proprietary interactive data mining system. This system requires low coding, enabling real-time visualization and interpretation across RNA, single-cell RNA, WGBS, protein, metabolomic, metagenomics data. That was my presentation. Thanks for your time and attention. Hello.
Thank you very much. Okay, so we have now got 10 minutes. So if anybody has got any questions, please feel free to either raise your hand, put it in the Q&A, or put it in the chat for our two commercial speakers today. Has anybody got any questions? Everybody's being very quiet today on this workshop. This is your opportunity to delve a little bit deeper as to what these different companies can offer you for your EV research. No, everybody's very quiet today. Is there anything further you two want to say about what you can offer, or are you happy that we move on? We have a question. Thank you.
Yes, hi everybody. I'm Hani Al-Shaika from the vet school, Nottingham University. I didn't join the meeting from the beginning, so I might have missed some information already. If we are interested in collaboration, how we can reach out to you and what are the logistics?
Yeah, so there's going to be a discussion at the end of the session this afternoon where we're going to talk about collaborations and what opportunities we've got available. So are you thinking collaboration with these companies, or are you thinking more broadly in terms of collaboration with getting into the EV field and doing transcriptomic analysis?
Yeah, both. I mean, we're open for all sorts of collaborations. We already work on exosomes. I have really extensive collaboration with King's College in London. So this fits well with what we wanted to do in the future.
Okay, so yeah, I'm sure both companies would be more than happy to speak to you about what they might be able to offer to help support your transcriptomic analysis of any exosomes that you've got. And like I say, at the end, we've got a 30-minute session when we're going to talk about what other areas people that are on this call might feel, what other useful collaborations that we could bring within the Midlands and wider afield.
Yeah, sounds great. Thank you.
Okay, thank you.
Cheers.
Alex.
Just to do a shameless plug, as you are Nottingham University based, we have the EV conference coming up on the 16th of April. So if you're not registered already, that would be a very good time to meet some of these companies and also to see the other researchers and students in the area. Shameless plug over.
Actually, having done that, Alex, could you please pop a link in the chat for people that might want to register for that event if they haven't already registered? If that's okay, please.
Absolutely.
Lovely, thanks, Alex. But yeah, that's obviously an in-person. This is just an afternoon workshop, and that's an in-person conference. Okay, well, thank you very much, both of you, for really interesting and informative talks. Thank you very much for keeping to time. So we will continue now with the next part of the session. So thank you. So Emmanuel from University of Leicester is going to talk to us about the challenges of doing transcriptomic analysis. Hi there.
Hello. Can you see my screen?
I can, yes. You put it onto slideshow. Is that what you were about to do? Fantastic. Yes, thank you very much. Right, I shall hand over to you. Thank you.
All right. Thank you so much for the opportunity to give some presentation on some of our work that we've been doing. So I'm Emmanuel. Currently, I'm a lecturer in cancer precision medicine at University of Leicester. Today, I'm going to give you some of the works that I did whilst I was a postdoc with one of our PhD students who did a very fantastic PhD work in extracellular vesicles. And whilst I was also interested in transcriptomics, so kind of like method optimization and also transcriptomic analysis. My research is mostly involving liquid biopsy, and at the moment, I specialize in circulating cancer biomarkers in breast cancer. So in liquid biopsy, mostly cancer biomarkers can appear in the blood by circulation due to active secretion or extravasation of the cancer cells into the blood. And whilst they are within the blood, when they die, they release a lot of DNA, RNA. And most of the time, the underlying cause is that we know that cancer can metastasize to different organs and it's through the blood. And we can actually take a blood sample from a cancer patient, and through that we can analyze whether the DNA, in terms of the circulating free DNA, or we can also do microRNA from the plasma. And at the same time, we can also do extracellular vesicles, which contains either DNA, RNA, or microRNA. And then we focus much more on CTC and immune cells, which is my main area of research, where I focus on circulating tumor cells within the cancer patients and also look at their immune profiling as well. But today, so that entails what liquid biopsy is. And it's not just blood. This is blood-based biomarker, liquid biopsy. You can actually get from pleural fluid, urine, and any other body fluid, where we can also isolate some of the body analytes and also do some analysis on them. But today I'll be focusing on extracellular vesicles for my presentation. So EVs are nanomembrane-bound particles, which are secreted by a lot of cells, mostly all cell types. And one of the things that they contain cargoes, which include RNA, DNA, and protein and lipid as well. And mostly the component that they contain reflect the cell of origin. Now, extracellular vesicles are grouped into three main groups based on either the particle size or either their method of release, mechanism, and the cargo that they actually contain. So when it comes to exosomes, they are cells, nanobound particles that are between 100 to 150 nanometers. And microvesicles are intermediate cells between 100 and 800 nanometers, and apoptotic bodies are bigger cells that are greater than 800 that are released by cells through other apoptosis. Now, one of the things is that in extracellular vesicles has been has mostly is elevated in cancer patients. So currently there's a lot of ongoing, where in liquid biopsy, we can analyze them and focus on whether to do tracking minimal residual disease using the molecular profiling, or can we use it for early screening, which is early detection, or molecular profiling, and at the same time, can we look at the components of extracellular vesicles and get a prognostic marker? Or can we use in terms of the analyte in extracellular vesicles for monitoring drug response or therapeutic resistance, which is still undergoing in a lot of the liquid biopsy research area. So in terms of isolation of extracellular vesicles, there's quite a lot of technologies that are available, approaches and techniques that can be used. Ultracentrifugation is the gold standard, which also we have precipitation-based method, we have immune affinity-based method, and ultrafiltration-based method. Now, for us, our goal was looking at EVs RNA, and one of the challenges when it comes to EVs RNA is that the method that you use can either be complex or time-intensive, and they are also instrument-dependent. And because we are interested in RNA, there's a lot of the amount of RNA that you get from EVs are actually very limited, and you can see from the early presentation that most of the RNA are a bit less than 5 nanogram. So we were much more interested in using commercial available kits, and one of the most significant, or let's say important, about commercial kits is that you try to avoid the issues that is associated with ultracentrifugation, which include protein contamination, low EV recovery, and the requirement of bulky or expensive equipment. So in this project, we're interested in two methods, which is ExoEasy and ExoGAG. Now, ExoEasy is a membrane-bound binding spin column-based technology that isolates exosomes and other extracellular vesicles. And ExoGAG, which is also a precipitation method, but it takes advantage of the negative charge glucose aminoglycans on the surface of EVs. And then once they bind with that, they will form a complex that can be purified by centrifugation. So when I joined my current group, we had a PhD student who was actually working on this method, comparing the ExoGAG and the ExoEasy. And I've actually put her PhD and her picture here because she did a lot of fantastic work for almost 5 years working on extracellular vesicles. You're happy to explore this, her PhD, which is online currently at the moment. That actually have a lot of information in there. And the basis of my transcriptomic analysis is based on the results that we got from her PhD. So the initial stage of optimization was based on cell culture, and so we use we use breast cancer cell lines and cell culture media where we grow them in FBS depleted cell culture media, and then while they are being passaged, we use the cell culture media to isolate EVs, but most of the time we go through several steps by doing centrifugation and also performing filtration using a 0.2 microfilter to remove more debris and also avoid some cells from it as well, and apply ExoGAG or ExoEZ to isolate RNA from the extracellular vesicles. So when she did her PhD and master's, she focused on using ultracentrifugation, extracellular ExoEZ and ExoGAG, and we're able to characterize them. So it is an image of an extracellular vesicles that we use the electron transmission microscopy to profile, and we realized that most of the RNA profiles that we get from the EVs that we isolate were less than, in terms of size, were less than 200 nucleotide. So we realized that most of the RNA, which is much more consistent with what we know, that EV's RNA predominantly are shorter and they are less than 200 nucleotides. So it will be best to look at the transcriptomics, or whether we can look at some RNA-seq from these samples. So that was not to go much more details into her project. One of the things also was that she did some optimization using these two methods, ExoGAG and ExoEZ, in healthy plasma samples. And the most interesting part was that when you look at the profiles here of some of the EVs that we got from the plasma samples using the nanosite to analyze them, most of the samples that we analyzed with we isolated with the ExoEZ had one peak, which is mostly around a median peak of 237 nanometers. But then the ExoGAG had two peaks, which sometimes peaks around 70 and sometimes also peaks around 237 or 197 nanometers, which shows that the ExoGAG gives a lot of variation of extracellular vesicles. And why we call them small extracellular vesicles is because we were using a 0.2 microfilter where we try to filter all the cells, any particles that is less than 200 nanometers. So most of the time we decided that, okay, we're gonna actually name them small extracellular vesicles, and also because we were going by the International Association of EVs based on their characterization and classification of how to name extracellular vesicles. We decided to stick with small extracellular vesicles based on the size that we observe on the nanosite. So one of the theories that we have was that the EVs that we isolated based on this the characterization, we thought a lot of them were exosome dense because of the size, because we are filtering them through a filter of 200 nanometer filter. So then we wanted to test whether there's a marker for exosomes where we use CD3 and a negative marker which we use CAN-X to investigate that based on their relative expression towards GAPDH. So what we found out was that there was expression of CD3 both in ExoGAG and ExoEZ, but there was higher expression in ExoGAG and negative expression of CAN-X using RT-PCR. Now, here, I just want to make the emphasis that we could have used, the reason why we didn't use protein analysis was because now with the ExoGAG, it has the reagent is much more purple in color. So once you try to, and which also blocks the fluorescence, so we decided to do PCR because the reaction that happens with the glycosaminoglycans and the particles has a purple color in it, which also will prevent downstream analysis when it comes to protein expression because of the fluorescence. So having been able to work out which of that we actually get some RNA from the extracellular vesicles that we isolate. The next thing was that we know that ExoGAR will give us more EVs, and also in terms of RNA isolation, we get higher concentration, mostly between 500 picogram as well. Now, and there were a previous study that have done EVs where in using this ExoGAR from endometrial cancers and demonstrate that EVs, exosome actually gives you a higher concentration of EVs and also a higher concentration of RNA. So I was working on AmpliSeq transcriptomics kit where we're using it for a different purpose, and we found out that we realized that we could actually, people have actually used this kit for obtaining transcriptomic data from highly formalin fixed paraffin embedded FFP, so FFP in short samples, and it's a kit that contains 20,000 genes in a single assay, and the input start from 500 picogram to 10 nanogram. And also we realized that we could modify the protocol based on this, based on the published paper that was actually done in, I think was published in 2019, that based on that we could modify it and we could also use it for lower RNA concentration input. So what I did was that, well, then we have to isolate some RNA from some cell line, and here we're interested in working on MCF7 cell lines and fervescent resistant cell lines. So MCF7 cell line, which has been developed to become resistant to fervescent. So we knew that we could use these two cell line to actually look at the expression to optimize our RNA input. So what we did is that we did a series of library where we start with 0.1 nanogram to 1 nanogram to 10 nanogram, because 10 nanogram is the standard input for is a standard RNA input for the kit. So we wanted to go down as low as 100 picogram to see whether if we modify the protocol based on the PCR reaction, can we get the same sequencing metrics compared to the standard protocol that the company have produced. So we make a lot of libraries. We sequence it on ion tolerance, which is an in-house. We have it in-house, so it allows us to modify the protocol and do a series of experiments to look at the different RNA concentration inputs. Now, what we found out is that when we make the library, before we put it on sequencing, we have to do quality check. But we realized that 1 nanogram input gives a consistent profile, and based on the library quality metric, you are supposed to get a peak around 300, an average base pair around 200, and 1 nanogram actually gave us a very consistent profile. So we realized that we could actually use 1 nanogram against the standard input, which is 10 nanogram. So we did that and we sequenced that, and as you can see here, this is the map read. And it produces an average map read of 12 million cells, 12 million reads per samples. And here we have comparing cells RNA, which is the 10 nanogram, and also which is the 1 nanogram. Now, once you use this kit, it has an in-house analysis, so it's already built in-house. And mostly you can see that we have an average of 95% alignment, which is much more higher. But when it comes to the target, so for example, target detected and valid read is mostly around above 90%, and the target, which is the percentage of genes that is mapped onto the ion transcriptomics target. So it's different target that has been put together as an assay, and we could find an average of 50% coverage when we sequence that. But then in terms of technical replicate, if you look here, you could see that the correlation among the samples where most of them were greater than 0.90%, which means that there's, which is a good replicate and much more better way to look at the data in terms of the different method that we use, using 1 nanogram as an input and 10 nanogram as an input as well. So we did some analysis where we used DSEP2 to look at differential express genes. And here we had a total of comparing the transcriptomics analysis between the sensitive MCS7 and the resistant fevaceren MCS7. We identified 1,574 genes, and we overlaid them between the two methods, and we realized that 27% of the genes are found between the two methods, and then there's independent genes that are found in different protocols. Now, here we are using a much more stringent analysis where if we lower the log2/4 change to, like, let's say 1, then we tend to get more genes in the 10 nanogram. But because we wanted to be much stringent about the protocol that we're using, we decided to stick with greater than 3, 4 chain, less than or greater than 3, 4 chain, minus -3, 4 chain, and a p-value of less than 0.05. So this also shows the volcano plot that shows the upregulation and the upregulation and downregulation of this the genes. And here we have the 1 nanogram, and here we have the 10 nanogram. And most of the time, the amount of cells, or let's say the genes that are found in the upregulations, are much more consistent within the two method. So one thing we're much more interested in, that which later on I'll state the reason why we're interested in the biological pathway, because they are resistant cell lines. We've already sequenced this on the Illumina, so we wanted to see, do we get the same biological pathways between the two methods? And based on the differential expression, sometimes some of the genes are highly expressed, or maybe the p-value is higher than within the two methods. We found out that they're still much more consistent. Just to highlight a few, we can see that this is the canonical Wnt signal pathway, which is found in the two method in terms of 1 nanogram and 10 nanogram, and which we're happy with it, because when we do in-depth analysis, we still find a higher correlation between the different biological pathway. So having established that we can actually have 1 nanogram as an input, and we know that we mostly get around 200 to 500 picogram in our EVs RNA, concerning our EVs RNA, we know that, well, 1 nanogram will be enough to perform some of the libraries as well. But we can't jump into plasma samples, but we are much more interested in we should test the EVs, culture them like previously that Tumi was actually working on, to see if we can actually get some EVs from the cell culture media even before we transition into the plasma sample. So we cultured the same cell lines that we've done our RNA analysis based on the 1 nanogram and the 10 nanogram. And then we use the cell culture media, filter them, centrifuge them before we subject them to exoGAG isolation, and then subject them to library, and then sequencing and do gene expression analysis. So this is just the quality metrics. The read was around a median around 6 million reads per samples, and we are using MCS7 and the fevaceren, and we can see here that consistently we have around 49% of coverage. And when we look at the sum between the technical replicates, the correlation here was around greater than 0.85%, which is much more still higher, even though lower than the normal cell RNA. So with this analysis, we only found 1,331 upregulated genes and downregulated genes, and there was no overlap. This a volcano plot showing the genes, whether they're upregulated or downregulated. And then this is just to show the first, so we rank them based on the significance. So the first 50 significant genes. And the most interesting part is that when it comes to those that are upregulated, majority of the first 50 based on their adjusted p-value. Majority of them were upregulated as well. But the most here wanted to look at the biological pathways, because now we've done biological pathways for the normal RNA, and wanted to see, do we get the same biological pathway when we do the EVs RNA from these same cell lines? And we realized that most of the upregulated and downregulated pathways were much more involved in antiviral and immune-related pathway, which including interferon gamma-1 signaling and lymphocyte activation. And when it comes to the downregulation, majority of them were sensory perception, amino acid and organic acid transport, and transmembrane transport. And one of the things is that these pathways that we are seeing are much more in concordance with some of the pathways being implicated in extracellular vesicle reaction. And also in the context of resistance in breast cancer, we can see that these are some of the pathways that are implicated in resistance. Now, one of the things that we were interested in, that because EVs is quite complex, we don't know whether the RNA that we have is still, there's some contamination of cellular RNA, or is the genes that we are actually seeing, are they really from extracellular vesicles or are they from cells? So we know that cells do not randomly dump RNA into extracellular vesicles. EV loading is very selective and regulated by cellular state. And we also know that not more of the EV RNA is equal to the whole cell RNA, but most of the time EV RNA is influenced by the changes in the cell RNA. So we test a hypothesis where we're looking at comparing the genes that we have identified within the extracellular vesicles RNA and the genes that we have within from the cells from the same cell line. And here is the 1 nanogram protocol, and you can see that independently here, you can see that for cells, we have 1,272 genes that are exclusive to just the cell lines. And here we have 1,029 cells that are very exclusive, very unique to the EVs RNA. And we only have just 320 genes that are found between the two RNA. And when we did, I did some analysis based on whether the genes that are common between the two analytes in terms of the RNA analyte, whether there's upregulation or downregulation. And you can see that majority of the time we have a correlation of 0.68 when it comes to looking at the 4-change correlation between the genes that are found between EVs RNA and the 1 nanogram, which is around a moderate around 0.8. Now, what this means is that we know that more than 1,000 genes are very specific to the EVs RNA, and it means that the EVs contain a signature that is different from the whole transcriptomics. And even though we know that we have 300, this is just a small portion of it, and even when it comes to the correlation, it's still moderate. So we did the same analysis, and we could see that in comparing the EVs RNA to the normal cell RNA that we have, even when we use the 10 nanogram, because there are two methods that we did, we still have low level of genes that are found between the two methods and the two different RNA analytes. So having established that, okay, we are much more confident that most of the RNA genes that we are actually getting are from extracellular vesicles, we proceed to look at them in plasma samples. So what we did is that we took some metastatic breast cancer patient samples where we've already stored their plasma samples, take an aliquot of that, filter it, and then perform an ExoGAG analysis on that, and also do some nanocyte analysis. So this is healthy control breast cancer patient and metastatic, and we can see that usually with ExoGAG, you're expecting 2 peaks, one which is much more less than 100 nanometers, and mostly around 200, and which is what we also saw it in the cell culture media, and in plasma samples we are seeing the same thing. And this is a freeze image analysis that we took from the nanocyte. So when we did the analysis, the RNA analysis from the small extracellular vesicles that we had from the metastatic breast cancer, we have upregulation of 165 genes and downregulation of the downregulation genes are around significant, around 83%. And this is just the heat map of the first 30 significant genes, and we can see that it's more variables when it comes to the patient samples compared to the healthy control. So what we did is that we just pick the upregulated genes and the downregulated genes and look at the some of the functions in terms of literature. So for example, if you take HLA-B, we know that it's been expressed overall in basal-like breast cancer patient, or studies have shown that. So we look individually for the work that has been done on these upregulated and downregulated genes, and we realize that majority of them have been implicated in exosomes or extracellular vesicles in the literature, which also gave us confidence that, well, some of the findings that we found in our RNA-seq in terms of the genes, we have evidence to back it that. For example, POS10, which has been involved in microRNA isolation biogenesis as well. So last but not the least, so the genes that we found, the differentially expressed genes, we put them on gProfiler, which is a platform that allows you to look at the biological processes or pathways of your significant differentially expressed genes, and we can see that most of the signals that we are getting are from regulation of signal transduction, intracellular regulation, and the platform also allow you to do not just the biological processes in terms of cellular, and also allow us to do molecular function, and also we have some microRNA coming through as well, and which probably we're not surprised, because when we did the EVs RNA isolation, we used a kit that can isolate both total RNA and microRNA as well. So in conclusion, we've demonstrated that we can actually get some limited RNA from extracellular vesicles, most importantly, the one we characterize as small extracellular vesicles. And using the implicit transcriptomics genes with the modified protocol, we know that we can actually sequence or analyze EV RNA. And we believe that if you combine these two technologies in terms of RNA analysis, it's likely that you can do more, we can identify some genes, not just for breast cancer, but also for other cancers where RNA quantities are very limited for gene signatures analysis. Now, one thing that with the kit, one of the limitation of this kit is that because it's negatively charged, and if you don't filter it, you are likely to isolate a lot of other particles that may not just be may not be extracellular vesicles. The other thing is also the mechanism, as I explained earlier on, that mostly you need protein analysis such as Western blotting or flow cytometry to actually verify whether your extracellular vesicles are actually extracellular vesicles by doing some protein analysis, which include a lysosomal marker like CD43. Even though we did this as a PCR form, the reason why we did it is because of the purple nature of the reaction that you get from the ExoGag. So one of the things that even currently the work that we are actually working on, we have actually decided not to use the ExoGag because we need to do more analysis to look at the confirm that we are actually having EVs, and we actually have Zuma like extracellular vesicles, and we realized that the ExoGag actually can't do that because of the purple reaction that you get from that. So I'd like to thank my lab and mostly to me, which initially have done a lot of method optimization, and later on Katie, who also came to join and currently working on EVs as well, where we've pivoted towards a different direction, looking at EVs, trying to try different method and move away from the ESOGAG as well. Thank you so much.
Okay, lovely. Thank you very much for very comprehensive talk. So I'm going to open it up to questions, and I see that there's one already being put in the chat. So I'll ask the first question for you if that's okay. So this is from Jessie, who said, thanks for the great talk. I'm a bit sceptical about the use of the kit to isolate EVs. They put it's true that you might have less EVs using ultracentrifugation or SEC, but the purity is much higher, and for transcriptomic analysis, it is a huge requirement. Your EV's characterization shows a heterogeneous population. However, the presence of calnexin confirms contamination and low purity in your samples. Are you sure that your downstream analysis is showing EV-associated RNA? Proper EV separation and characterization is essential to perform correct high throughput analysis.
Yeah, so I agree with everything that the person is saying. That is why even at the moment, we knew that there's a limitation when it comes to the ESOGAG because of its reaction and how the heterogeneous populations that we were getting. Now, the only way we could be much more confident with the gene expression or downstream is what we're trying to use bioinformatic analysis to compare what are some of the pathways that are actually coming through when we look at the EVs, the downstream analysis. So I think there's a bigger limitation when it comes to the ESOGAG. And to be frank, we've actually even tried new method and we've moved away from the ESOGAG.
Yeah, I was going to say there are some concerns with using that and the purity that you're getting and potential contaminations. And I think that that's why we're having this workshop, is to talk about what works and maybe what isn't maybe such a great technique to use going forward. So yeah, thanks for the question, Jessie, and for your good answer there. So there's someone with a hand up. Would you like to ask your question verbally?
Yeah, hello, Emmanuel. Lovely presentation. But it's just to carry on with what Jesse said, because I was actually asking myself why you guys didn't characterize the extracellular vesicles that much, because the only thing I could see was the transitional electron microscopy. And then, but you mentioned it at the end anyway about Western blots and flow cytometry and the rest of them. So, but is there a reason why you guys didn't bother to do it initially? Like, or even check about the what's it called? I don't know if you've seen this paper by I think Professor Clayton and Professor Webb about checking the ratio of particles to proteins. I think it was written in 2011 or thereabout. So I was wondering why you guys didn't do that initially.
Okay, so I focus so much on the transcriptomics aspect because that's why I put the PhD thesis in there for people to go and read, because there's quite a field of characterization that was done that I didn't have much time to put. I think that would be so much to present. Yeah.
Okay, no problem.
Yeah.
I think it's really great discussion. This is why this is why we've organised this workshop. You know, the challenges with working with extracellular vesicles, I think is hugely underestimated by anybody coming into this field for the first time. You know, to get pure populations, to be able to do the downstream analysis, and then on top of that, to work out what those changes might mean and what biological context that it might have is a huge challenge in the field, and one that I think we just all need to keep working together to try and do the best that we can in a very challenging situation.
Yeah, I totally agree. I think I started this field probably 2 years, and I've seen a lot of changes that has happened when it even comes to the method. And every single time, the International Association of EVs are always coming up with new characterization that the field is actually evolving. So, which is good for this discussion.
Yeah, no, definitely. I mean, I like you, I've been in this field for about 5 years now, and every time you look back, you realise you've got to think about a new control. But that's great, and that's really incredibly important. Right, has anybody else got any further questions for Emmanuel before we go on to the break? No? Okay, great. Well, I'm going to post the agenda up so you can all see that we're going to have a short break now. But please stay on the line. Just leave your camera and your microphone switched off. Go and grab a coffee and a biscuit. We'll be back at 3 o'clock sharp, please, because we've got Professor Vicky James Midlands, from the University of Nottingham, that's going to think about, what actually means when we get this extracellular vesicle microRNA data. What does that actually mean in terms of biological context? So Vicky is going to come up at 3 o'clock and give a 30-minute talk. And then at the very end, we're going to have half an hour where we can talk about, what the challenges are, what we think we need to do as a group, and as individuals interested in extracellular vesicles and specifically transcriptomic analysis of extracellular vesicles, how we can help and support each other in this field. So please go grab a coffee, tea, whatever you drink you like, and we'll see you back here at 3 o'clock. But please do stay on the call. Thank you. See you shortly. Hello and welcome back, everybody. So we're going to start the next session, where I'm going to invite Professor Vicky James to talk about extracellular vesicle microRNAs and how to take it to understanding the functional of this data. So Vicky, I'll hand over to you if that's okay.
Thank you very much, Lucy. Can you see my slides okay?
I can, yes, thank you.
Brilliant. So yeah, I get the task of, I guess, trying to make sense of some of what we do with microRNAs and how we make some kind of functional interpretations of them. And I'm sorry to disappoint everybody from the beginning, but I don't think I have many of the answers. And what I've put together today is probably more of the questions and more of the observations that have just really cropped up over a number of years of working in this field. So just for a bit of a background on me, most people classify me as a vesicle biologist. But actually my background is in non-coding RNA and particularly microRNAs from the start. So I probably come from a slightly different perspective to people that come into this field from the EV biology side of things. So I think for me, one of the key questions really is, do the EVs package microRNAs to be functional and to mediate that intercellular communication, or are they mostly a byproduct of RNA turnover? And that's one of the questions that's really plagued us when we've been looking at these extracellular vesicles and how they mediate communication. So today I wanted to have a little look at what some of the evidence was and why the field has been shaped the way it has. So the first question in addressing that is, are these microRNAs selectively loaded? Are they stochastically loaded? And what's the evidence around either of those options? So one of the first things that I wanted to mention as well is, are we actually measuring the wrong pool of RNAs? And this is something that came up from the last talk as well. And it's whether these RNAs actually really are vesicle encapsulated, or whether they just co-precipitate with vesicles. And I know in the EV field, there's so many arguments about the different techniques that you use. You should use size exclusion chromatography because it prevents this. You should use centrifugation because you get this. You shouldn't use precipitation techniques because of contamination. But actually, when you get down to it and you really experimentally look at where different RNAs are stored, there is no single technique that will eradicate all these types of contamination and guarantee that you have only got vesicle RNA there. So it is really worthwhile thinking about and testing your protocols out to try and find out where your RNAs are based. And I'll come back to that again a little bit later. But this whole question about selective loading versus waste disposal. So we know that you get an enrichment of microRNAs in cells and that they go up and down with different stimuli. And cells do have to get rid of these microRNAs at some point in time. So it is questionable whether EVs are a route to actually manage cellular homeostasis and export some of these unwanted RNA species. And that might be one reason why we see some differences between sort of exosomes versus the other small vesicles that capture up some of that cytoplasmic element of the cell and just export it out that way. But there's certainly some evidence that really strongly suggests that some microRNAs and some non-coding RNA species are selectively packaged. So we see that they have different motifs. They're linked to very specific RNA binding proteins. They are actually sorted, and often their sorting occurs under different types of stress conditions, which just go an extra step to suggesting that their export is selective and it is dependent on the physiological state of the cell at that particular time. And this really does argue against random dispersal. I think waste in general is rarely this regulated. But I think we probably have a combination of both. There probably are some RNAs in our vesicles that are just there from a waste management point of view. But then I think there are also some of these selected ones. And we have to kind of think about what microRNAs are we looking at and what mechanism of them being in those vesicles is likely to be the most realistic. And this brings us on to our next problem, and it's a problem that's been in the field now for a really long time, so probably well over a decade, and this is the idea of stoichiometry. So many studies estimate that actually when you look at your EVs, you've probably got less than one copy of a specific microRNA per EV on average, and that really begs the question, is that enough? How many EVs realistically fuse with a recipient cell? How many copies of the microRNA will they actually be transferring? Is that enough for that microRNA to be loaded into a RISC complex and be functional? If the dose is really below a certain threshold, can it really be biologically meaningful? But then there are some studies that come out that have really countered that argument, and that's to say that actually one copy per EV is an average across your heterogeneous populations. If you could subpopulate your sample size, so you only picked a population of vesicles that you were really interested in, they might be highly enriched in your microRNA of interest. And it might be that subpopulation of EVs that's actually getting into your recipient cell type. So they might actually be delivering quite a concentrated dose. You just can't see it when you do bulk RNA analysis because of that evening out across those heterogeneous populations. And there's also some cellular context which add to this as well. So the local microenvironment. So we know things like synapses, particular tumor niches, they can achieve really high concentrations of EV uptake. So again, that really does rock that contradiction there of how many copies do you need in an EV for it actually to be biologically important. And we also need to take into effect repeated uptake over time. So although many of us look at our studies in vitro, so we might just give one dose of EVs, in reality within an organism, there's that repeated accumulation over time. So that dose of microRNAs might be small within the EVs themselves, but actually over time they might be building up and accumulating within a cell under certain conditions. So I think it's probably good to bear in mind that we do need to consider whether there are enough microRNA copies for it to be biologically meaningful, but also that biology doesn't operate on these population averages and it does operate on effective local doses, so it's very context dependent. So why do we not have the answers to these questions? Why has the field really been struggling to get a grip on this? And that's really because most positive studies rely on overexpression of microRNAs in those EV donor cells or artificially loading microRNAs into these EVs. And that leads us to have high EV doses, which we can then use quite nicely within our in vitro studies. And you completely understand why people take this approach. It's very hard to show with small microRNA concentrations that you've got any biological change. But what we're really demonstrating here is the possibility that that's the mechanism, and we're not really demonstrating physiological relevance. So we're questioning if we're actually really studying the biology here, or if we're actually engineering an artificial signal that we're then putting on in vitro and trying to interpret something from it. But evidence is emerging, and more recent studies are using sorry, someone just took control of my slides. People are using sort of RNA tracking technologies. So you can label up microRNAs, you can label up other RNA types, and you can use tracking mechanisms to see whether they're actually getting into the cells. And if they are getting into the cells, whereabouts in the cell are they going? So, for example, are they loading themselves into processing bodies where lots of microRNAs are stored? Can you co-localize with RISC complexes? And studies are also looking at so phenotypic loss. So if you have EVs that are depleted in these microRNAs, do you lose the phenotypic difference that you were seeing before? So while not universal, this type of data is quite microRNA specific at the moment, but it does give us some support to show that these microRNAs can actually be functional. And this is just an image here that's come from my group, where we have had a go at doing some RNA tracking within vesicles to show that it is possible to do. But there's still problems with the data, and the problems with the data really come from that lack of having a direct mechanism. So we all know that EVs carry a number of biologically active cargo. So we know they carry proteins, lipids, RNAs, DNAs, they've got cytokines in them, proteins on the surface. The way they interact with different cells is different. The way they get into the cells is different. So there's all sorts of different ways in which the cargo of an EV can change the phenotype of the cell. It isn't necessarily microRNA cargo. It could be protein signaling. We could be looking at TLR activation, surface ligand receptor interactions. There's a whole host of different ways that it can occur. And the other thing that's not really taken into account when we're looking at the microRNA cargo of EVs as well is that when you actually put these onto your recipient cell, your recipient cell has its own version of RNA regulation within it. It won't necessarily just accept some exogenous RNA coming in and act upon it and do functional changes. It's got its own mechanisms of RNA regulation, which will take these things into account. So these studies aren't always that easy to try and interpret and show that direct effect between an EV microRNA and a change in your cell type of choice. So that leaves us with a bit of a quandary. So what do we do? So when making predictions, it's really important that we consider the cell or the tissue context. So what usually happens is we get our EV microRNA cargo data. The first stop that we'll go to is a microRNA target database, so something like TargetScan or MirBase, and we'll have a look at all the different genes that might be regulated by those microRNAs that we found or that we're interested in within our cargo. We'll then take that, we'll pop it into a pathway enrichment analysis, and we'll have a look at what pathways come out of there, and then we'll try and speculate on the effects that these EV microRNAs might have, and then perhaps we'll pick a few microRNAs for further study. Problem with this approach is it completely ignores any cell type or mechanistic context whatsoever. The good news is some of these databases, particularly the pathway enrichment databases, are getting better, and there are some out there where you can add in this cell type or tissue type of context, and you can ask it to have a look at different types of mechanistic or signaling areas. But in general, if you're just using this sort of default global approach, it is really ignoring both your tissue or cell type context and any mechanisms that you might really be interested in from a biological point of view. So why does this approach not always work? And I've got an example here on the right-hand side of the slide. It is unfortunately a real example, and it's a bit of an extreme one, but we see this coming up quite often. So this was that there was pathway enrichment analysis, and it showed that this particular microRNA 1513P was a cargo in lung cancer EVs, and it was predicted to target the CATSPA signaling pathway in localized lung stromal environment, and it had a really great p-value, looked excellent sort of on your pathway predictions. Problem is, for anybody who's good on their sperm cell biology, CATSPA signaling is in sperm cells. It's not something that happens in the stromal cell biology cell type. So again, that's just the risk factor that if we're not really focusing in on the context and the biology, that you can draw these really spurious speculations and then go down these rabbit holes of looking at specific microRNAs, which really perhaps should have been discounted at a much earlier stage. So if we're thinking about cell type context, we can pick microRNA targets with knowledge of what the recipient cell actually expresses. So many of us will have RNA sequencing data sets, proteomics data sets of our cell types of interest, our recipient cell types. So we know really what those cells express under their normal growth conditions or under particular stress conditions or pathogenic conditions that we're interested in. And this can be a bit of a guide to knowing which microRNA gene interactions are the ones that we might really want to focus on. And as I mentioned before, pathway enrichment can be tailored as well. So that can be put into the context of your cell type or your tissue type. And again, you can start to think about the genes and the microRNA combinations that go into that pathway enrichment to get something that's a bit more biologically meaningful. And just a note as well, we've found as well when we're picking between our microRNAs and their gene targets, and we're using some of these databases, it's a good idea just to have a look at those default settings, because often they will pick out pretty much everything, and that might be where you've got a scientifically proven microRNA target, so an experimental evidence base that microRNA does regulate that gene target, all the way through to completely speculative predictive targeting, and the two are very different in terms of their probabilities. So if you want to be really cautious, you might want to pick really microRNAs and their gene targets where there's really a good experimental evidence for them, or that their prediction ratios are really stringent. Other questions to think about as well that we sort of ask ourselves when we're setting up some of these biological experiments is, are we looking for a local signal, or are we looking for more of a long distance signaling effect? And what's the actual mechanism that we're interested in? Because sometimes there's a rare mechanism, it's contextually critical for your biological system that you're looking in or in your model, and p-values become less important and they can mask some of that biological relevance. So I think it's important maybe not to always go on a fishing trip and just say, I found all these microRNAs, now I need to find the targets and I need to find a mechanism. but what's the mechanism that's actually underpinning your phenotype or your biological question that you're really interested in? So once we've picked our biologically relevant microRNA and we've thought about our potential target or potential mechanism that we were interested in looking at, what do we do next? What's those next sort of experimental steps that we could plan? So one of the first things that we've often done with our experiments is to test whether our microRNA is really inside the EV. So we've done this with some really quite simple experiments, and that's just using some RNase treatment. So we can take our RNA samples, we can treat with RNAs, and we can see whether our microRNA of interest is still present. Can we still pick it up by qPCR, for example? We can also extend those to use detergents, so bursting the EVs and then adding in the digest step to see whether we then can eradicate the microRNA of interest, just to give us a little bit of reassurance about whether our RNA is within the vesicle itself. That's not a completely foolproof mechanism of doing it. There are other ways of looking at it. You can use sort of immunoprecipitation or sort of magnetic beads to pull out different vesicles and see whether the RNAs correspond with those vesicles that you pull out. But this is just one approach that we've used and we've found to be quite successful. So then it comes down to what type of evidence can we gather to show that our EV microRNAs have some level of biological function. So often there's quite weak evidence associated with that, and that'll be detection of the microRNA in the EV itself by doing some validation experiments and then correlating with some phenotype, so putting those EVs on a recipient cell type and then seeing that perhaps that mechanism we were expected to change does in fact change. But really, I think the field's now moving forward to getting that stronger line of evidence. So really demonstrating that that microRNA is transferred into the cell. So using some of those RNA tracking techniques, that actually the microRNA itself is loaded into a RISC complex or comes loaded in a RISC complex. So it has the potential to be functional in the recipient cell. And then using sort of reporter assays to look at target repression. Now, there's some people that would argue that a reporter assay is rather artificial. So again, you're not showing that physiological relevance. But if you're trying to look at an mRNA within the cell that's usually there, you're also battling against that cell's own RNA regulation processes. So it can become a little bit more complex to, again, show that direct relationship. Whereas with reporters, you can have your microRNA target site, you can also mutate that target site, and you can show that actually your vesicle-delivered RNA has an effect there. And again, you can use some of these loss of function or rescue type experiments or depleting the RNA within your donor cell to show that when you put your EV on, actually it's the EV microRNAs that's doing the job. So this is just a couple of examples of techniques that we've used in the lab to show you that they do actually work. So although these aren't microRNAs here, these are messenger RNAs, we've used different tagging techniques to tag the RNA within our vesicles, then deliver it to recipient cells, and then pull back out that tagged RNA from those recipient cells just to see whether firstly we can see it and how long that RNA really sticks about for within the recipient cell types. So that's one way that we can prove that RNA cargoes are actually delivered into the recipient cells. And then here on the right-hand side, this is just an example of a reporter assay that we put together so we could see whether these delivered microRNAs were having effect on our reporters. And in both instances, we could see that actually when you have the EVs delivering the microRNAs, you see further repression of the reporter assay to different extremes, but we could see evidence that that was actually happening. So just a couple of examples of sort of work that we've been able to do. So I think ultimately for EV microRNAs to be biologically important, what we are ultimately trying to prove is that there is quantitative sufficiency. So there is enough of that microRNA there to be effective, and that might either be by the number of copies of the microRNA loaded into the vesicles, or the fact that you've got a subpopulation of vesicles that's concentrated in the RNA, or the context allows those vesicles to be concentrated, so for example at a synapse. Is there selective loaded or stimuli-driven loaded of the microRNAs, or is it just random? So is it again that these have a biological importance, or is it kind of just more that they're in there because they're being exported as part of a waste process? Is there mechanistic delivery of the microRNA? So is there at least the potential that the microRNA can be functional? and then that demonstrable physiological consequence that microRNA does directly change something. And I think now if any of these fail, the biological significance probably is questionable for that microRNA. So that brings me back really to the close of my presentation, and it brings me back to that key question again of, are EVs packaging these microRNAs functionally, and are these mediators of intracellular communication? I would say perhaps, but probably not always. So microRNAs have never really been dominant regulators in the cell RNA biology context. They've always been fine-tuners of gene expression. So I think to expect them to be dominant regulators because they come from an EV might be stretching the possibilities of what they're capable of. But I think they are context-dependent modulators. So with particular physiological relevance in particular niches or particular cell types or tissue types, or in different disease settings or stress stimuli, certainly I think they do have a role in modulating how cells behave and what they can do. So I think the real question that we should probably be asking ourselves is whether EV microRNA signaling, not whether EV microRNA signaling exists, but where and when it actually matters and what those specific microRNAs might be within our own model systems or our own disease context. And with that, I'll just put up an acknowledgement slide because obviously there's plenty of people in my group that do lots and lots of the work, and I'd be very happy to take any questions.
Lovely, thank you very much, Vicky. Excellent talk and also very thought-provoking, so thank you very much. Are there any questions either on the chat or if anyone wants to put their hand up to ask Vicky any questions? So I think for me and the work that we do in my lab, we have obviously looked at microRNAs, and we've also looked at downstream, how it modulates function. And I think the point you make in the sort of biological context, you're normally not just going to get exposed to the EVs once, you're going to get exposed to EVs several times. And the data that we've just published actually shows that it has to have at least 3 lots of EVs added to the cells to actually cause any downstream impact. So I guess my question is, you know, what would you recommend as a almost gold standard way of seeing whether you think any of those microRNAs are actually having an impact or might have a biological relevance?
That's a really excellent question, and I don't have an answer for you to that one. So there are studies out there that have a look sort of at in vivo release rates and uptake rates to give you sort of an idea of perhaps how many vesicles might be in the circulation around a tissue at any one time, to sort of give that idea of what a recipient cell might be receiving, say over an hour or over a day. But in different disease contexts, it changes. So in cancer, it changes massively because they kick out loads of EVs all of the time. So their concentration is really high. In different stimuli, it changes again. In normal homeostasis, it's probably quite low. So I think really you have to have a look in your context, have some sort of idea what might be happening within our own body systems, and then try and replicate that in some way. But I think it comes back down to trying to keep those doses as physiologically relevant as possible without like totally overloading what we put into the cells, which I think is historically what we've always done to try and see that outcome in the cells. So sorry, I don't think I can answer your question on that one.
No, but I think it's just this is what the whole idea of this session is to sort of make us think about what, you know, depending on the model systems that we're using in our labs, which is the best way to try and prove the best that we possibly can that maybe these microRNAs are actually having or have the potential to have an impact. Thanks, Vicky. Has anybody else got any questions? No. Okay, well, thank you very much for your talk.
There is one in the Q&A, Lucy.
Oh, thank you. I didn't see that. Thanks, Georgie. Do you want to read it out for me? I can't see.
Yeah, so Tanvi said how to narrow down gene targets for microRNA from DEG microRNA data.
Yeah, that's always quite a difficult one to do. So like I said, you can go to the databases and you can see what genes would your microRNA target, but there's lots of things to take into account there. So the first being most genes are targeted by more than one microRNA. And actually, if you look back at microRNA biology, they have to have the effect of multiple microRNAs binding at the same time in order to get repression. So it's worth thinking that, do you have all of the microRNAs to target that gene? Do you only have one? Do you have 3 of the 5? And which ones are important? And then the second thing is, again, how has that database decided that that gene is a target of that microRNA? How confident would you be in the prediction? So if there's experimental evidence that, yes, that microRNA will target that gene, you can have a good degree of confidence and probably include it in your list. And then you need to work down that order of probability. The next is, how good is the seed site sequence? How good is the probability that it will bind within the 3' UTR, and there's not other factors that are going to prevent that binding, which comes down to mRNA structures. And you can get probability scores for all these things. So it depends on how comfortable you feel with those probability scores as to what you want to include in your list of those genes that are targeted by your microRNAs. But again, it comes back to really understanding the microRNA biology and how they actually bind to the 3' UTR, how they have to act in concert, and how the structure of the 3' UTR changes that binding. So there's not a simple answer, but my advice would be, don't just export it on the default, have a really good look through and question, how would they actually work? What's the biology? Are these genes expressed in my recipient cell type? And try and work your list that way.
Great, thanks, Vicky.